the KTH for an institute in the electrical department. This is a multidisciplinary group, so there are some from biology, some electrical engineers, and diverse engineers. The title of this thesis is called electrophoresis and electrowetting as an anti-Foley process for antibacterial surface. First, on the outline, I'm going to introduce the theme, then the background where this thesis is based, then the experimental setup where I assemble a device which contains the design, chip fabrication, and the gasket mount. So now there are some measurements and results, and numerical simulations and experimental tests that were done on that, and at the end conclusions. So as an introduction, we are focused on bacteria on the surface. We have seen that in the history, bacteria has led into infections, so we can see that on the prosthetics. In inner tubes, that's like endotracheal tube, we can see that there is also a kit of cleaning for the devices. And in catheters, we can see that there is also a cleaning that can deteriorate the lifetime of the material of these catheters. In the hospitals, we can see that there's many sections as infrastructure, also cleaning areas, personnel hired, and research on the area. They are more focused on what is focused on the bacteria that is in the surfaces and the equipment for sterilize the bacteria. So at the end, we have the bacteria on the surface. The main purpose of this thesis is to get away the bacteria from the surfaces without attaching. As a first time if the bacteria attach, it's really difficult to deattach. So how can we dispose of the bacteria? Now, some background to take account. There are two particles that have ions in an ion aqueous medium. So here we are going to talk about what is the electrical double layer. If we can see, we can have a zoom on it. We can see that this is the wall of the particle. There are some ions, and here is the medium. So as soon as there is the contact of the counter ions of the ions, there are going to be a screening that is called the Weyl screening, and it's not uniform. So they are going to have a layer on top of it, and there's going to also be a counter ion layer. This two is called the Stern layer, and the other is called the Hansel plane. Now we are going to introduce the DLDO theory, where in the energy versus distance graph, we have the attraction potential, and also we have a repulsion potential. The attraction potential is due to the van der Waals forces, and the repulsion is mainly by the Karolinska repulsion and also the medium. So if we zoom up these two pictures, we can have this picture that is the energy surface versus the distance. And we can see the more closer to the distance, you need more forces to pass the energy. As we increase the electric in the medium, there's going to annul the repulsion potential, and we are going to have similar to the attraction potential. The mechanism of the bacteria to attach is they have a mobility at the beginning, then they have a diffusion, which it is really easy because if you apply some forces on the bacteria, they can be repelled. As soon as they attach to the surface, the range of forces are really high, so it's going to be really difficult to get detached. So our point is just to keep the bacteria to not attach and with really low forces. This is a picture of very known bacteria as E. coli and Dendritin, and we can see that very close to the surface they can attach. As part of the theory, we have electric field that we are going to use. So if we have a particle in a uniform electric field, there are going to be ions on the particles, and there is going to be a dipole moment that is going to be formed. This dipole moment is going to make a force that is going to be attracted to this pole, and in the same way in the other direction. This is called electrophoresis. What happens if we reduce one of the poles? In this case, the electric field is not uniform anymore, so there is a gradient of the electric, and we have a lower field and a higher field. So in this case, the force is not going to be the same anymore, and we have a higher force. This force can be depicted as this formula, and in here we can see what is called the Clausius-Mosolti factor, which only depends on the particle, the electricity, and the medium, the electricity. And this is the base of the electrophoresis. We can simplify this formula by approximating the particles as a cylindrical or a spherical, and we obtain this formula, where we always are working with complex dielectric permittivity. So as we can see, the porosity of the particle only depends on the particle and the medium and the relationship of it. So if the real part of the Clausius-Mosolti is higher than 0, the particle is going to move into the higher field, and this is called positive electrophoresis. On the other hand, if it's negative, the particle is going to move into the lower field, and it's called negative electrophoresis. For simplify this, we are only going to talk about attraction and repel from the electrode to the particle. Another theory to consider is we have a droplet of water, we have an electrode inside of the droplet, and we have on the bottom another electrode. If we apply some potential, there's going to be a change on the surface energy, and the droplet is going to change the contact angle. There's also a wire opening where there's no contact with the droplet, as in the first place, and it is called white coring. The Lippmann-Jung equation describes this behavior. As you can see, there is the angle that is moved by the voltage, the original angle, the capacitance between the medium and the electrode, and the surface tension of the liquid and gas. So we can see also that the voltage is up to the square, so it's really sensitive to that. This is called electrowetting. But if we cover, as we can see on the picture of the ito, we cover with a Teflon layer, is going to call electrowetting on the electric, because we add a resistance on it. So now we have the theory to assemble the device. First, we design. The project idea is to have electrodes that can affect the bacteria on the gasket, and the bacteria can be repelled at all the time without being attached. And we can create an antibacterial surface with that. So we have to consider a wafer with electrodes and a gasket with a fixed hole diameter, and then it's going to be attached to it. And we have to consider that it must be sealed, so there were no medium to be poured out. So the electrode designs, I have been doing a research, and there are different configurations of the electrodes, and there are from the rotation particles to the accumulating of the particles between the electrophoresis. So the most similar to our purposes are this one. So what I have done is I made different measurements of the electrodes, and I made a table where I have 3 cases with different measurements of the electrodes and a fixed length of the electrode. This electrode is going to be inside a cell. So I have 6 cells on the top that are active and 3 that are not connected, and that's a reference. How I done the chip fabrication? I have a wafer of 5 microns of thickness. I put metal first, titanium tungsten to have adhesion to the glass wafer, because it's a glass, has to be transparent, and then gold. And after that, I have a photoresistor for the lithography. So at the end, we have these layers. As we can see, this is the wafer, this is the metal that has been sputtered, and this is the photoresist. I have used a mask for the photoresist, so I apply UV light into the mask. As you can see, this mask has patterns on it, and all the UV light is going to enter only by the holes of the patterns. And the green light is the hardener of the photoresist. Then I will wash away with an etching process where I etch all the gold and all the photoresist that is not being cured. And at the end, I made a method that is called stripping, where the photoresist went away. So at the end, I have only the glass wafer with the metal. And this is the result. Then I see that they use, in the commercial way, in the labs of the biological, they have these culture plates. So what I have done is measure these holes and to mimic this and make it easier to the biological persons that are helping me with in Stockholm. I designed a basket with the same characteristics, with the whole diameter of this, and I made 3 samples, one for the 9 hole that is going to be, and 2 for tuning the same material. So I have one version that is called with a material of stoichiometry kaolin. There is a new material in the market, has a patent in Stockholm and in US. They are working with them. And I've made different molds of it. And after that, I have seen that there is no uniform because there are different curing times for the polymer. So I have did different holes with different measurements just to tune the exact time and to have a really nice and uniform gasket. Then I made a gasket version 2 where I used PDMS, which was really practical and fast. And I only used one whole gasket, and the sealing on the Teflon was really good, even though the characteristics were not as accurate as the Oscar. So these are the results of the base that I have used for the polymers. And this is the final device I have did with soldering the electrodes. Then the measurements and the results. I have simulated for the electrophoresis. So this is the bacteria. As we model it in the electrophoresis, we have 3 layers: the cell wall, the membrane, and the cytoplasm. Each of these layers have different conductivities and permittivities. So what I have done is I use the mirror of the sphere approximation. To make an approximation, I have a model of the bacteria. So with this is a complex model. I use MATLAB to depict this. So we have the frequency that we are using and the real part of the chromosome sorting. We are interested only in the repulsion of the bacteria. So we are going to use only low frequency. I have simulated on console program, and as we can see, there is an E. coli at different voltages. And the reaction is almost the same, but the magnitude is different. So what I have done is I enter into the distance of the electrodes, and at 37 micrometers between an electrode of 8 micrometers, I have found that the forces are really high. So I can reduce the weight of the electrodes to have a more levitance of the bacteria from the surface. I also have measured the length of the diffusion of the bacteria, and with the Einstein small cross relation with the kinetic theory, I have the mobility. The mobility is in direct proportion with the force, that the only force that is mainly applying to the bacteria is the electrophoresis force. I also made tests on electrowetting on dielectric, so we can see the same configuration. And this is the setup I have made with a camera to measure the contact angle of the droplets that I have shown. First, I need to see if the evaporation of the droplet has some effect, just to not be that biased by it. And I can see that after 400 seconds, there's effect. So if I can do the measurements between that period, I have not the effect of the droplet evaporation. As you can see, the first is without voltage, and this is with the voltage. I made with different mediums, as PVA, water, and a medium for the bacteria, and the black line is the equation of the theory. So it really goes different from the theory. And the most detailed is 30 volts. So I made bacteria tests on Karolinska Institute in Stockholm. We put with measurements in different directions. We wash out the chip, and then we look at it in the microscope to see how many bacteria were attached. And the chip, at the first moment, were no bacteria attached at 30 minutes. Then I put Teflon on glass slides, and after 30 minutes, there were no bacteria attached neither. But after 24 hours, we see some bacteria attached to the glass. While the voltage was active, I see some electrophoresis in the bacteria, as you can see there, a line. And also negative electrophoresis, as you can see only in the electrodes and not in the other part. Some of the problems was the breaking of the Teflon because it was really thin. And also electrolysis in the medium because of this break. So conclusion is a study of the electrokinetic microenvironments that we were aiming to, and it was a really success to enter into these devices. And it's a step forward to looking into the bacterial surfaces. The BDMS gasket resulted to be a good sealing in the chip, even with the Teflon, that this was really hydrophobic. The MATLAB complemented really well to the constant simulation. And the electrowetting on the electric, it was really time dependent, as I discovered, because the longer the time I put a droplet, the higher the voltage is needed to have a contact angle difference. Future chip improvements are being looking for as etching processes, because we have done another chip with better etching, and we are ready to make more experiments with that. Preference? Thank you. That's
a wide presentation on the topic. I would like to know which has been your personal activity in the whole project.
Actually, I have made a lot of research in all the topics, because at the beginning they were told me you need to enter into this. I had my background in electronics, but I have never touched this kind of things or topics. So I did a really deep research, and they are working different projects. So I have to do this by my own, almost. And they were satisfied with the results, as they can. One of the supervisors that I have is one of the membership of the MEMS conferences around the world, and he presented that on a conference in the Stockholm University. So I really have to get on deep on the electrophoresis and the contact angle measurements by myself, doing also the software research for that. Was not easy, but I never have entered into the nano systems. That is really, really small world, and there are different considerations I have to take. As an electronical engineer, I was more into the macro level, and this is more in the micro. So it was a huge challenge, and I liked it.
Thanks a lot. Very good.
Really?
Congratulations.
Congratulations to you. presentation. It hasn't come from me. Something.
per dire